All right. I appreciate everyone coming today. My name is Kurt Veely. I am a biostatistician with Berry Consultants and the Director of Education for the Innovative Clinical Trials resource here. I want to talk today about an introduction to innovative trials. This is our first of several webinars, and I'm going to give an overview and essentially a kind of a whirlwind tour of the types of innovations that are available. I'm going to start with a brief history, not a lot, but some notion of why we're at the place we are. I'm going to talk about why gold standard randomized clinical trial is a standard. I'm going to talk about the components, the purpose of each, and the potential cost of breaking these. Whenever we talk about innovation, we're really talking about deviating from this gold standard. And so what are the trade-offs involved in that? What are the types of innovations, and why should we use them, or why should we avoid them in certain contexts? So modern randomized clinical trials, they emerged in the 1940s. There's a trial for patulin with the common cold in 1943. It looks a lot like a modern trial, but it doesn't have randomization. The real first modern clinical trial is the streptomycin trial in 1946. And what it adds, kind of the last component here, is randomization. And so it looks like a modern trial: randomization, blinding, controls, all the things that we think about. And you can actually read this paper in 1948. It's still publicly available. So where are we in this? So if we think about the science of experimentation, this 1946 trial has a number of principles that are at some level timeless, notions of randomization that we definitely want to keep. And in a lot of fields, we see things like the fundamentals of calculus or chemistry, where what we do today looks a lot like was done 100, 200, 300 years ago. At the same time, fields also advance. And here are some examples of other things that were going on in 1946. So this is a computer. This is ENIAC in 1946. Obviously has a lot of the fundamentals of computing, but really doesn't look like a modern computer. In this room, for example, now fits easily on your desktop, much smaller than that. This is an operating theater in 1946. Certainly seems quite Spartan compared to something we would do today. I was going to have a blank slide for mobile phones, but I actually found out that there were mobile phones in 1946 based on walkie-talkies. Difficulty with the 1946 mobile phone is only 3 people in a city could use them at the same time, and that was restrictions on how the frequency space was used. So certainly we've made, in many fields, huge advancements since 1946, and we've done that in experimentation, in clinical trials as well. So I want to talk about, for innovation, this standard randomized clinical trial that existed in 1946 and we use today, it certainly performs well. We're going to be talking about innovations, about deviations from this space. And so I want to talk about, again, what are the features in the gold standard, what are the negative aspects of the gold standard, and what are the trade-offs from a variety of innovations, really getting at the notion of what parts of this 1946 trial are timeless and we want to keep, and what parts can we do better on that we've improved over time. So let's start with the components of our gold standard. The first set, you can see in the center, it says we want to avoid biases. So we want to get inferences that we think don't have confounding variables and other problems. The ways we address that, we include stratification. Stratification is intended to avoid known confounders. If we know men and women are going to be different in a trial, we try to make sure we directly equalize the arms on men and women. We may equalize for sites or geographic regions. We don't know all of the possible confounders, so we also randomize. And so this accounts for unknown confounders. And essentially, anything weird that might happen in the trial, randomization gives us a way of breaking that confounding. So we've got stratification for known confounders, randomization for unknown confounders. We also employ blinding. This is, or masking. This is a notion to avoid subject and assessor biases, avoid the placebo effect, avoid the fact that an assessor knows who got the treatment and the control, and they might assess patients differently. And I've also got this notion of a fixed sample size. We don't want to just allow an experimenter to ad hoc, well, I'm going to experiment until my treatment looks good, and then I'm going to stop and go, hey, look, it looks good. So we incorporate this fixed sample size where we say we're going to go to 500 patients, and this is going to give us our answer. And again, that's a bias mechanism to avoid random highs, random lows. Other components of our gold standard are aimed at obtaining really interpretable, conclusive results. So one reason we employ a two-arm trial in many cases is it's very nice at the end of the trial, this is what we've changed. We've randomized patients to control or treatment, and any difference that we see, we'd like to attribute it. It's either you got control or you got treatment. Differences between arms, that's what they're due to. This is the cause. We also typically employ a homogeneous population. We spend a lot of time before the trial going, who are we going to do this experiment in? Again, this is aimed at really trying to focus where we think the drug will work, a device will work, whatever we're working on, and we want to, at the end, be able to say, hey, for this group of people, we have a conclusive answer. We usually employ standard validated clinical endpoints, notions like mortality, things that we know if we move this, we have made a difference in patients' lives. And I've also put a fixed sample size on this slide as well, and this gets at the notion of this conclusiveness. We want to make our trials large enough so when they're done, we feel we have enough data to answer the question. So we want to make sure our trials are adequately powered, and part of adequately powering them is this controlling the notion of a false positive at the same time. So the trials are large enough to produce an answer. This entire structure does phenomenally well. It does what it says. It avoids biases. It gives us clean, interpretable answers. It also comes with some major limitations. In particular, it is resource intensive. There's a lot of components to it. Typically, it requires a large sample size, and a sample size that can be infeasible in rare diseases. It also requires us to often ask narrowly focused questions. I think we've all had the experience of sitting around planning a trial going, should I look at this population or that population? Boy, it would be nice if I could look at these individuals, but the drug might not work there. I don't want to risk it. All of these things ask us we got to pick a population. We've got to pick a dose. We've got to pick a treatment effect to pick a sample size. All of these things are often at odds with the uncertainty that we bring into a trial. And by really trying to narrow our focus down, we may run into a trial that very admirably answers a narrow question, but it doesn't answer our real question or questions. The other aspect of this is also create a societal prisoner's dilemma, which I'm going to talk a little bit more about in later slides. But the notion that if we run multiple clinical trials, if I'm running 30 or 40 clinical trials in Alzheimer's or heart disease or something else, each one of those can be optimal for its task. But if we look at the collection, we get some odd behavior. And again, I'll come back to this. So I'm going to go through a variety of innovations. All of them are aimed at addressing those limitations. For each one of them, I'm going to give a quick description over what they do and a notion of the trade-offs. So let's start with the minimizing of resources. We have a resource-intensive trial. Is there any way to do this with fewer resources? So the first notion available is to have a flexible sample size. So we're breaking this. Remember, fixed sample size appeared twice in my gold standard. So a flexible sample size is implemented. We conduct one or more interim analyses, pre-specified times. You got to write all this out in advance. At each one of those interims, we can stop for futility or success. We can change the sample size of the trial. Examples of this include futility stopping, a group sequential design. This is a formal mechanism for stopping early for success. A Goldilocks design. This is similar to group sequentials. It allows for incomplete data. Or a sample size re-estimation. So we can look at the data early and go, no, 500. We were going to go to 500, but now that we look earlier, we realize the variance isn't right or something's not right, and we think we're going to need to go to 800 subjects. So we're going to change the sample size. So all of these break this fixed sample size, but they do it in a way that is intended if the question is answered or the question changes, we're allowed to make adjustments. The kind of gains that you get from a flexible sample size, it does what's advertised. It reduces the expected sample size of the trial. It makes it smaller. It can make it decently smaller, especially futility stopping, probably the most efficient mechanism of what I've talked about. If the trial doesn't look like it's going to win, you can stop early, and often you could stop, say, 50% of the patients or more. This also accounts for uncertainty in the treatment effect. We've had trials where people have come in and go, I've got this early phase 2 data. The treatment effect might be anywhere from a value that would require 50 patients to a value that would require 200. And so that's a broad range. You don't want to just pick one of the extremes and end up with a problem. So we can account for this uncertainty in the treatment effect by incorporating a flexible sample size into this. You get the benefit if it's big, treatment effect's big, you can stop at 50. If it's small, you can continue. The losses of this is, remember what we were using a fixed sample size for. We were trying to avoid multiplicities of allowing the investigator to stop ad hoc. So we have to account for the multiple analyses. They have to be pre-specified. We have to make sure we've done it in a way that there aren't any multiplicity biases that are associated with it. Part of that adjustment is often the maximal sample size may have to increase. It gets a little bit bigger, not much, but it gets a little bit bigger, and so this compensates a bit for the expected sample size getting lower. You may also, if you stop very early, you may have limited information about, say, subgroups. If you were planning on running a 300-patient trial, you stop at 150, there's less information about all the groups than you might have otherwise anticipated. You may get less information there. There's also potential concern about statistical biases, which you can estimate in the planning phase. Another mechanism for minimizing resources is to do single-arm trials. Don't enroll a control. This is breaking another aspect of our gold standard. This is really acting as information being brought into the trial. If you run a single arm, you have to go out and get a historical data set, and you end up comparing your current treatment subjects to that historical data. And so that's common in rare diseases, usually based on prior clinical trials, a registry, case studies, some group of patients with your rare disease. The advantages to single-arm trials are, of course, they decrease sample size. You don't have a control arm, so that's 50% of your subjects saved right there. You also have the advantage that in a population that's averse to getting placebo or the control, you aren't giving it, so it's potentially easier to accrue. The losses here are we need a historical estimate to compare against. So there are significant costs if that historical data set is inaccurate. If we're enrolling a population that is healthier or sicker than what we're comparing to, we may walk in with the game effectively rigged one way or the other, and we're going to get inaccurate results. We also have broken with a single-arm trial. We can't blind anymore. Everybody knows they're on treatment. So all the biases associated with that come into a play in a single-arm trial. A mechanism for avoiding some of those risks with a single-arm trial is historical borrowing, different than a single-arm trial. Here we augment the control arm with historical data. There's a graphic at the bottom. So we take historical controls and combine them with a smaller number of current controls, and then we compare them to treatment patients. So by reducing the control arm and augmenting it, we save that sample size, but we're not saving the entire control arm. This retains randomization, it retains blinding, and in addition, we get some verification of our hypothesis that the historical information was on point, because we can compare the current controls to the historical controls. The benefits and risks look a lot like single-arm trials, but they're mitigated here. So the sample size benefits are smaller but go in the same direction. We also potentially find it easier to accrue. We need a historical data set, again, and the other aspect here is while we can test our assumption that the current controls and the historical controls match, if that assumption isn't valid, there's not really much of a backup plan here. Essentially what you have to do, if the historical data wasn't on point, you get the benefit of knowing that that's true, but you're then going to have to run the larger trial in order to actually get a full answer there. So that was minimizing resources. We can also use innovative trials to ask broader questions. And so this is often people only think of innovative trials, can I do it cheaper? And this is a case for broader questions. We're really trying to do it better. We're trying to answer something that would not ordinarily be answerable. So a standard randomized clinical trial, it requires a lot of focus. We have to do one specific treatment arm, usually. Some multiple arm trials are pretty common. We have to specify a population. We got to power for a treatment effect. And usually this notion of looking at one arm, we only look at one domain. So if there are multiple, if a treatment is multimodal, we may only look about one of those domains at once. And we don't look at combinations. All of those things are typically absent. So this focus generates a notion of anticipated regret. This notion that you should think at the end, if I fail in this trial, what am I going to think? Am I like, okay, my question's answered, or am I going to go, boy, I wish I had done the trial in this patient population, or I wish I had picked a different dose, or I wish I had just run a larger trial. All of those things are anticipated regret. So innovative trials, they aim at answering this full question or set of questions. And so we're really trying to get more robust trials, and we're trying to do it in a way that has a minimal amount of increased cost. But we are talking increased cost. I found this quote in another project. This is from R.A. Fisher, a very famous statistician, in 1926. And I think this is kind of the crux of my message here. He says that no question is more frequently repeated than we must ask nature few questions, or one question at a time. So again, this notion of focus. The writers convinced this view is wholly mistaken. Nature will respond best to a logical and well thought out questionnaire. If you have multiple questions, if there's multiple aspects, you should design an experiment that asks all of them. If we ask her, nature, a single question, she will often refuse to answer until some other topic has been discussed. So again, this notion of anticipated regret, this other topic that you're going to have to deal with. Examples of trials that incorporate broader questions. Factorial designs, this is a very old kind of design, but it's used fairly minimally in randomized clinical trials. These are trials aimed at exploring multiple domains. So instead of, say, one trial where you're exploring factor A, you might do big A versus little a, and another trial with factor B, big B versus little b. We go ahead and put them all together. So we do A and B together, A without B, B without A, and neither one of them at the same time. These can generalize. So we have a trial example where we tried to vary ventilator, antibiotic, and steroid administration. So the graphic at the bottom, this is a possible trial where there are 3 ventilator strategies, V1, V2, V3, a couple antibiotic strategies, A1, A2, and a couple steroid strategies, S1, S2, somewhat generic. But the idea here is that generates 12 arms. And so I've got all the 12 arms running within one single trial. So this often there's a notion of, oh, that's a big trial. It turns out that factorials are always better than running separate trials in each one of for each variable, for each factor. And how they're better depends on how the factors work with each other. If the factors interact, That's a situation where the effect of one of the variables depends on the settings of the others. If that's true, then only the joint trial can find it. There's no other way if you do separate trials and you do one factor at a time, you often will not see the interaction because you don't fully explore the space. If the factors don't interact, then you get another benefit. This is a very nice mathematical property that's somewhat not intuitive, but if the factors don't interact, they're additive, the effect of A is always the same no matter what happens at the other settings, then you can estimate the effects of all the factors for a similar cost as exploring just one of them. And so that seems bizarre. It's like, I get extra domains for free, but that is effectively what you get. The information about factor 1 for each patient answers the question about factor 1. The information from factor 2 answers the question about factor 2. You really basically get 3 times as much data for each subject if you're exploring 3 different domains. So factorial designs, again, this is the only way to detect interactions. It explicitly allows for combinations. Without interactions, additional domains are almost free. So this is a case, I've tried to keep the gains and losses to have the same number of boxes and try not to tip the scales. If you really want to investigate multiple designs, this is a no-brainer, or multiple domains, this is a no-brainer, this is what you want to do. The losses here, they can be a little harder to plan. You've got to consider all the different domains at once in planning. And certainly, if interactions are on the table, if you want to estimate them, you're going to need a larger trial. But keep in mind, if interactions are on the table, there's no other way to do this. You can't run at a time smaller trials and estimate the interactions. So broader questions, dose ranging designs, I think these are more common. I think a lot of people are familiar with these. A lot of modern trials have 1 to 2 doses in them. We often make guesses at what the 1 to 2 are. We may pick, somebody else ran this, so I'm just going to keep the same dose, or I pick a couple values. And a lot of times the motivation for that is, if I picked more doses, I'd have to have a really, really big trial. A dose-ranging trial typically incorporates multiple doses, 3 to 6. You may do more than that. We've certainly had trials that have 15 doses or continuous dosing. And the way these are handled is usually a dose-response model is incorporated for efficiency. So you try to combine the information across multiple doses to give yourself an estimate of a common curve. And usually we incorporate some kind of adaptive features in this, which as the trial continues, it focuses, it eliminates allocation to poorly performing doses, and it increases allocation to doses that are performing well. The net result of all this is to try to get extra doses in the trial at minimal cost. The gains here, we certainly have a better chance of having the correct dose in the trial. If you can put 6 doses in the trial, the odds are better the right one is one of the 6 than if you only put in 2. In addition, you have a better chance of finding it because you can actually focus your study as you go. And as I said, with modern modeling and adaptation, the marginal cost of the 3rd, 4th, 5th, 6th dose can be mitigated significantly. So if you want to have if you've got a trial that has 2 doses in it, taking it up to 4 doses by no means doubles the size of the trial. Eventually, these extra doses, it's a minimal amount of cost. The losses here is, while the increase is mitigated, it certainly is an increased cost. So you will have to, in the planning phase, figure out what that number actually is. And in addition, the assumption of dose response, that assumes a curve. They're often robust, but if they're wrong, you can have problems. And ways they might be wrong, you might assume monotonicity and find out your dose response isn't monotonic. Those kind of errors can happen. A final broader question here is an enrichment design. So all of these are aimed at our clinical uncertainty going in. Enrichment is aimed at the notion of I have to pick a population in advance, all these inclusion-exclusion criteria. So what happens if I'm uncomfortable with that? I think, hey, maybe I'll start with a certain population, but it's possible the drug really doesn't work in part of this, and maybe some of the patients that aren't in here, maybe it does work well. An enrichment trial directly addresses this. So an enrichment trial is aimed at starting with a specific population, and as the trial continues, you can either remove part of that population, you assess at an interim analysis, it's not working, we're going to not enroll these kind of patients anymore, or you can add to the population that you're enrolling. Hey, it worked really well for this group. What about a group, it worked for a group that was this sick. What about a group that's just a little bit sicker? Maybe I should add those as well and see if it works. So you can see the graphic here. This is the evolution. You start with an initial trial population, and as the trial continues, maybe you remove a little bit of that population, and I ended up adding a little bit of population as well. So the final selected subset represents an evolution from the initial trial population. Examples of this, you can see this in oncology. This is where a lot of these have been pioneered in terms of what's called basket trials. These are often targeted agents that are being tested in multiple tumor types. So you may be interested in something that expresses BRAF, and you test that in breast cancer, ovarian cancer, lung cancer, all of these expressing the target. And as the trial goes, you can eliminate certain tumor types as the trial continues. We've also seen these trials in stroke, where we're interested in enrichment to determine the time window and the stroke severity where the treatment is effective. So how long can you wait before administering this treatment and still get a benefit, and how severe a stroke is there of value to doing this? The general gains and losses for an enrichment trial, certainly the main gain is exactly what it says. It allows us to identify the right subpopulation. And usually it does this in a way, we know that if we do a regular trial and we do a whole bunch of post hoc subgroup analyses, those are prone to error. This is a way that allows us to continually, we make adjustments to our population, and the design itself, it confirms those decisions because it continues to enroll. So this is robust to our initial choice of population. So if you don't really know your initial population, this is potentially a great choice. The losses here is because you have multiple groups, because you have adaptive features, you're adding and subtracting groups of people, you're going to have a complex statistical methodology to keep track of that. You need to make sure at the end of the day you're going to get the right population or something very close to it, and you're not going to be making a lot of errors. So that requires a lot of statistical expertise. The trial typically has to be larger than doing something homogeneous because you have to allow the possibility of all these groups. On the plus side, that may actually allow you to enroll faster. So if I'm enrolling a broader population, that potentially gives me a better opportunity to get patients in because I have more patients eligible. All right, so that was broader questions. I now want to switch gears to societal considerations. Talk about Alzheimer's. There have been a lot of failed trials in Alzheimer's, at least 25. Depends on how you count. They all had different sample sizes. Just for simplicity here, I'm going to assume that they were all 1,000 per arm. As a society, what have we done? We've enrolled 25,000 control subjects, and we've enrolled 1,000 subjects each on 25 experimental therapies. Now, each one of those trials made sense. They were one-to-one. They were a gold standard randomized clinical trial. As a society, we've enrolled 50,000 patients, and we've done it in the way on this slide. We have tons and tons and tons of controls, and we've got 1,000 on each of our 25 experimental arms. If we were doing this as a society, if somebody came to us and said, we have 25 experimental arms, we want you to investigate all of them, we would never randomize this way. It is way too many subjects on control. We know tons about Alzheimer's always has a bad progression. We know the time course. We know all of this information, and we really have extra information here. We don't need 25,000 allocated to control. What a platform trial does is it's a mechanism to handle this societal issue. We want to randomize between multiple experimental arms. So we want to be able to run all of these 25 treatments through a single trial. They're not going to all happen at the same time, so a platform trial allows arms to enter and exit. So this may only be investigating 4 or 5 treatments at a particular time, but these 25 trials occurred over multiple years, so arms enter, arms leave. One of the key aspects for resources here is the control allocation can be reduced, and that reflects the sharing of control between the arms. So what we're going to be able to do is instead of allocating 25,000 control subjects, we're going to share that information. Instead of just having treatment 1 is only compared to the 1,000 controls in experiment 1, we're going to allow it to use all of the controls. This kind of mechanism, lots of talks and papers on this, but the societal savings can reach 30 to 50%. And some examples of this, there's the paper at the bottom by Woodcock and Levange talking about in New England Journal of Medicine. It gives examples of these in oncology, antibiotics, Ebola, Alzheimer's, and antipsychotics. So this has been done, it's been implemented, and been implemented successfully. The result of this is we end up with a randomization that looks like this. We're essentially going to save our control subjects. That lets us either run fewer subjects through, or we could add more treatments. If there has been a treatment 26, 27, 28, 29 that never got investigated because we didn't have resources to do it, there are resources that are now available that we could have done it with. So it just depends on how you want to, in effect, spin the savings here. But this seems like a much more efficient allocation for society as a whole. So platform trials in general, some of the advantages here, really as a society, there are massive savings compared to running separate trials. There are operational advantages of having what's usually called a warm base. If you have a new treatment coming in, you don't have to set up site agreements and IRBs and set up randomizations and all the other stuff like that. The general trial has taken care of this, usually through something called a master protocol. And what you're able to do is you do a minimal amount of work in order to add this new arm into the trial. So new agents, the startup is far faster. The losses here is this is a very complex animal. These are new, and it takes a lot of effort to get everything balanced for arms coming in and out and make sure they work. So all of these, it is harder to start, harder to get going, but once it's going, it gets a tremendous amount of momentum. It also, of course, requires multiple parties to cooperate. Those 25 different treatments, they didn't run identical trials. They had their own quirks in each one of them, and you have to get everyone to agree to work in the same sandbox. And so that can be awkward. But keep in mind, I mean, right now what we're doing as a society is people come together to investigate a particular treatment for Alzheimer's, and we have multiple investigators on grants. This platform trial idea is getting at the notion of people should come together to investigate Alzheimer's, and then we can bring lots of different treatments to the table. So this was a set of innovations. There's also a set of tools that you can add essentially to most of these innovations. One of them is these are adaptive features in terms of changing randomization. This is aimed at efficiently focusing our resources, and I'm applying this to multiple arm trials, not a 2-arm trial, but these are trials like factorials, dose ranging, or platforms. We typically achieve this through arm dropping, which means if an arm is doing bad, we just get rid of it, or adaptive randomization. And adaptive randomization means instead of just completely getting rid of an arm, we're going to reduce the allocation to some arms a little bit, and we're going to increase the allocation to other arms a bit in proportion to how they're doing. So the graphic at the bottom, you can see over the course of a trial, we may start with equal allocation to a control in 4 arms, and by the end of the trial, we've got more subjects on control. We may increase that over time. Arms 1 and 3 are gone, and really the trial is trying to figure out whether it likes 4 or 2, and by the end it's really decided maybe 4 is the right answer, so that's what's getting most of the patients. The advantages to this is it decreases the sample size. You're eliminating arms, directly saving there. The focus itself also increases your chance of identifying the best arm. So being able to focus, if arms 3 and 4 look to be the best, being able to focus on arms 3 or 4 gives us a better chance of identifying which one is actually the best. You can also avoid patient exposure to poorly performing arms. That's usually a benefit. We often refer to the top box here as benefits to the external patient population and also to the bottom to the internal patient population. The losses here, this requires careful calibration to avoid premature early decisions, and it can create operational complexity. So certainly you have to keep into account that different arms can, as they come in and out, you've got to handle drug supply and so forth. Another possible tool that's valuable here is to use more informative endpoints. We're actually having another webinar on this topic. There are many endpoints that result in smaller trials, biomarkers or surrogates. In rare diseases, disease progression models, they're particularly valuable in rare diseases. So this is a way to get as much information as possible out of every subject. We also have subject-specific endpoints, like, and by subject here I mean the area, the indication. So ideas like hospital-free days, where we are penalized to give 0 for mortality. So we essentially say, when do you get out of the hospital, but if you die, you don't get any credit for being out of the hospital. Those kind of measurements are usually more sensitive than mortality. There are also endpoints which synthesize information, utility measures which combine safety and efficacy, for example. All of these things are aimed at allowing for smaller trials. So we can have another hard endpoint as a secondary endpoint, but all of these are aimed at, can I get the answer with fewer subjects? The endpoint may also directly measure the mechanism of action. In a phase 2, we often use a biomarker because that's what we're interested in phase 2. The losses here is, this is the notion this innovative endpoint can create interpretation or acceptance issues. If you change from a standard endpoint and you get a successful trial, people may go, well, what am I supposed to make of that? Is that necessarily a good thing or not? And also endpoints like biomarkers, they may not have an effect that translates into something clinical. So you always want to be careful about making a decision on an endpoint and then being overly confident that that's going to translate into a broader trial. So all of these things are intended to help us. As a summary of what we've talked about today, the real idea here is to try to use innovation to make trials either smaller or better. Those aren't necessarily at the same time, but they can be. We want to be able to essentially answer the real question that's been asked. And each of these innovations presents its own set of trade-offs, and you need to plan for these and go, in my setting, is this something I want to do, or is this something that I don't want to do? Over the course of the next year, 2018, 2019, we are going to be running a number of additional webinars. These are going to include topics that we have discussed in this webinar today. Obviously, I've given you a whirlwind tour. I haven't given you all the details of any of these. Just trying to give you what's on the menu. The additional webinars are going to concern multiple arm trials with arm dropping, innovation in endpoint selection, so that will include, for example, disease progression model in rare diseases. We're going to have some examples of how those would work, and also flexible sample sizes. We're also going to incorporate 4 short videos. Those are single-arm trials, subset selection, so this is related to enrichment, safety and efficacy in a utility, and futility stopping, which is obviously incorporated into flexible sample sizes, but really aimed at how do we monitor for futility and really make sure that we get out of trials that aren't going on a good path. If you want to contact us, our contact information, we have a public website, the innovativeclinicaltrial.org website. This has a blog and other features, and we'll gradually obtain more content throughout the year. You can also email us. Dore Sitko is our project manager. Her email address is there. My name is Kurt Veely again, and my email address is on the screen. We certainly welcome questions and comments. In giving this webinar, we've tried to leave a lot of time for questions at the end. So certainly, any questions that you have, please ask them now. And also, if you have other questions, part of the purpose of this public website, we're going to have a blog on there. If you have a broader question, can you tell me more about a particular topic? If we get a question, we certainly intend on producing blog entries and other materials to try to explain that, and that will depend on public interest. So again, I appreciate your time.